Welcome to another lecture by MedicoMedics, Pharmacology Chapter 4, Drug Development and Regulation. In this lecture, we will briefly discuss drug development, talk about drug discovery, preclinical testing, clinical trials, the process from drug development to market release, and end with a summary. Drug development is a long and carefully regulated process aimed at bringing safe and effective medications to patients. It begins with drug discovery, where researchers identify and optimize compounds that have potential therapeutic benefit. Once a promising compound is found, it undergoes preclinical testing. This includes lab experiments, animal studies, all to evaluate safety, toxicity, and biological activity. If preclinical data is favorable, the drug then moves into clinical trials, which are conducted in phases. So phase 1, for example, tests safety and dosage in healthy volunteers. Phase 2 assesses efficacy and side effects in a small group of patients. Phase 3 confirms effectiveness in a larger population. Now, some drugs also undergo Phase 4, or post-marketing surveillance, once they're on the market. Finally, all data is compiled and submitted for regulatory approval to agencies such as, for example, the FDA in the US. Now, these agencies review the evidence to ensure that the drug is both safe and effective for clinical use. Only after approval can the drug be prescribed to patients. And this structured process ensures that therapies are scientifically validated before reaching the public. Now, let's start by diving a bit deeper into understanding the initial stage, which was drug discovery. Now then, drug discovery is the first critical step in the development of new medications. See, drugs can come from several sources. Some are natural products derived from plants, animals, or microbes. Penicillin is a classic example, originally discovered from a mold. Others are synthetic compounds created entirely in laboratories, for example, aspirin, and it's developed through chemical synthesis. However, a growing class of modern therapies are biologics. These are derived from living organisms. These include complex molecules like monoclonal antibodies used in cancer and autoimmune diseases. Now, the process begins with target identification. Scientists search for a specific molecule involved in a disease, such as a receptor or enzyme that the drug can act on. Now, once a target is established, the next step is screening. This involves testing thousands of compounds in automated systems to see which ones interact with the target in a desirable way. From there, a few promising candidates move forward for further development. Now, preclinical testing is the stage that follows drug discovery, and it serves as a crucial checkpoint before a drug can be tested in humans. The primary goal here is to evaluate the safety, efficacy, and pharmacokinetics of the drug through laboratory and animal studies. Now, several tests are conducted during this phase. We begin with toxicology studies. They are designed to assess potential harmful effects, including organ damage, carcinogenicity, or reproductive toxicity, etc. Then we do pharmacokinetic studies to examine how the drug is absorbed, distributed, metabolized, and excreted. In our previous chapter, we dove into greater details on pharmacokinetics and pharmacodynamics, so make sure you go and repeat. Now, these steps here help determine dosing and frequency. In addition, efficacy studies are conducted using disease models in animals to evaluate whether the drug produces the intended therapeutic effect. Importantly, preclinical testing is a regulatory requirement. See, a drug must demonstrate sufficient safety and biological activity in these studies before it is allowed to move into clinical trials involving humans. And this phase helps filter out unsafe or ineffective compounds early in the process, reducing risks in later stages. After preclinical success, the drug moves to clinical trials in humans. So once a drug has passed the preclinical testing, it enters the clinical trial phase. This is where it is tested in humans to evaluate its safety, appropriate dosage, and efficacy. And clinical trials are conducted in a stepwise manner across 4 phases, each with a distinct purpose. So in phase 1, which involves a small group of healthy volunteers, and here we focus on safety and dosage. Researchers look for side effects and determine how the drug is behaving in the human body. Phase 2 expands the study to a larger group of patients who actually have the condition that the drug is intended to treat. The main goal here is to assess efficacy and continue monitoring for side effects. Then we have phase 3. This includes an even larger and more diverse population. This phase is designed to confirm the drug's effectiveness, comparing it to existing treatments, and to monitor for adverse reactions across various patient groups. Finally, phase 4, also known as post-marketing surveillance, it occurs after the drug is approved and on the market. It tracks long-term safety and effectiveness in the general population. It helps to identify rare or delayed side effects. Now, each phase builds upon the previous one, creating a comprehensive understanding of the drug before and after it reaches patients. Now, let's look at the regulatory approval process that follows successful clinical trials. Now, after clinical trials, a drug must undergo regulatory approval before it can be prescribed. So agencies like the FDA in the US or the EMA in Europe review a new drug application, or NDA, which includes all preclinical and clinical data. They assess the drug's safety, efficacy, and manufacturing quality. And the outcome may be full approval, a conditional approval, or even a rejection. But even after approval, the process doesn't end. See, the drug enters post-marketing surveillance, where ongoing monitoring, known as pharmacovigilance, tracks adverse effects in the real world. This helps catch rare or long-term side effects. It is also important to note the time and cost involved. See, on average, it takes between 10 to 15 years and everything from $1 to $2 billion to bring a single drug to market. So these high figures reflect the need for extensive testing, complex regulations, and the reality that only 1 in perhaps 5,000 compounds ultimately succeed. So in summary then, the stages of development include drug discovery, preclinical testing, clinical trials, regulatory approval, and post-marketing surveillance. Drugs undergo rigorous testing for safety and efficacy. Regulatory agencies ensure that drugs meet standards before and after approval. And that's the end of this chapter. Continue now to chapter 5.